Okay, recap on 3D comparison. We can describe proteins on the level of 3D. So this is the actual 3D structure, the coordinates of an atom. In 2D, which is the distance map. And I said that 2D, the information content of 2D is almost entirely containing the 3D structure, except for its mirror image. And then there's a representation on 1D. And that reduction from 3D to 2D doesn't make a big difference, but from 2D to 1D completely changes it. So now we no longer really know the structure. This is a simplification. That still contains some features. And a lot of what we will discuss is about those features in 1D. I talked about the comparison of sequences. The simplest one is you move visually this the sequence around here. You try this, this, and that, and essentially you try all possibilities. And the way you do it, and I'm going to use this recap here to go through these slides again. Hopefully it becomes more clear this time. So again, what I show is the one protein, the other protein, their sequences, and the ones in this matrix are indicating that in that alignment position you have a hit, a match. Match is defined as identical letter. And what you see here is the complete matrix of all the ones. So these are all the matching letters you find in that matrix. Okay? So that's the first step. You build up the matrix of all ones. In the next step, you have to move through that matrix. And the move you can do at every single position. You can either go in the diagonal, so when you begin the G on G, which gives you a score of 1, you can move along the diagonal and take the next one here and the next one here. So you would do GQ against GG, right? And the score of these would just be 1, because there's only one match so far. You could also go into a gap state. So you leave one of these out. But if you Let's look at the first problem here where we don't allow gaps. And the problem where we don't allow gaps, all we can do is move on the diagonal. And what I show here is actually the sum of all the hits, which is 1, because you're not having a second hit. Okay? In a different, if I started with this G here, then upon the second one, I have a hit. So GQ is, we now have matched 2 letters. We go further, we do not have an additional hit. Okay, again, we can move on the diagonal, and if we moved on the diagonal, the sum of all the hits at the end remains 2. It was as high here as it is here. And again, I can only move on the diagonal because in the simplest model, I do not allow for gaps. Not allowing for gaps, I have to align the next in plus 1 and j plus 1. That is just on the diagonal, okay? So much. Now is that part clear for everyone? And by the way, somebody asked whether the slides, so I was told that the slides were online last Tuesday, and they should be online now. Yeah? No, he just ran away. But typically what we promise, no, is, you saw the Dimitri who was standing here, I showed the photos of him. Complain to him, but he needs a week, and none of them are online yet. It is true that Game of Thrones kills our free time at the moment, and there's a very high probability of messing it up in our unit at the moment. So I apologize for that. We will try better, but talk to him. Maybe when you talk nicely to him, maybe he's going to be more motivated. I mean, again, so he does a lot. It's not. Okay, but again, what we try to do with the videos is that they are there within a week. We can the PDF should be there before the lecture, and please remind me when that is not the case. Okay, so without gap, it looks like that. Now, what if we allowed gaps? So in the language before, allowing gaps means that rather than going from here, aligning the V to the A and next aligning the E to the K, you allow yourself to not align the E. Or, sorry, not align the K. So put a gap in here, and instead, so sort of skip the K, and instead take the E to E alignment. And now you add up to 3. And you do another gap, and you add up to 4. And get this alignment, yeah?
But how can you be sure that if you do make creates a gap at the E that you don't end up with more points the other way.
How can you make sure that what you add up is still a good score?
Yeah.
So yes, that is a simplification, and that in particular for the example that I show here, you may remember, I'll get to that in 2 slides again. So the probability that you have in single gaps, so gap, letter, or something like that is very unlikely. So the example that I show here is a little bit misleading. Okay, I would have to spend more time on this one. But ultimately, the answer to your question, how can you make sure that what you do is right, I believe is on one of my next slides. Give me a sec for that. It's already on this one here, where the score for the entire thing is essentially the match, there's a delta function, times some penalty that you give for a gap, and times the number of gaps. So minus that. So now this is still not answering your question, because this is clear. This comes out of my algorithm, this comes out of my algorithm, but the open parameter, the free parameter here is the gap penalty. So how much do I penalize introducing a gap? And ultimately, your question is answered by exactly that choice. How do I choose that? Let's just say we choose it empirically. So we look at a bunch of alignments and we see what is a typical gap penalty that helps us to reproduce what we believe is right. Is that then answering your question? Then ask again, then I didn't understand your question.
If you continue the diagonal, how can you be sure that you don't get more points?
That you don't get other?
Get more points this way.
So you mean in this one? No, you do them all. I'm sorry. Okay. So this is the one that is still not right in my example. I showed you in the first matrix I showed you all possible ones. I'm showing here you only one possible path. But you essentially follow up all of them. Which, going back into my matrix here, and into the original one matrix, you see there is not that much. Which in fact, the whole matrix is relatively sparse. Which is not necessarily a typical example of this example, because essentially you have 20 amino acids. So the match rate is 1 in 20. So essentially in a typical matrix, only 1 in 20 of the numbers are filled. So this is not untypical. Okay? That means you can relatively easily follow up through this. Okay? Relatively easily, you don't even have to do a computation. It's an if, not a plus, so you even save time on that. But you guys are not. So when I learned programming, then there was a difference between if and plus. Today, no longer, right?
Could you one more time explain the formula? What is the G?
The G is essentially the gap penalty. And why there is an O to it, I get to in a minute. But essentially, at this point here, you insert a gap, one gap. N is the number of gaps. In this particular example, I have 2 gaps. So it would be 2 times the gap open penalty.
What is the penalty?
So that's the penalty, that's a value, it's a free parameter that you have to optimize, and you can, for instance, optimize that by making sure that the type of alignments that you know are right, you actually get. So you fit the parameter on data. That's a heuristic way. There's no theoretical way in which you can do it, but it's something, obviously, a biological meaning of letter, 100 gaps, letter, is nil. So at some point it's clear that that gap penalty would be too small, and then there are gap penalties that are too large, which is when it doesn't allow a gap ultimately. In the other extreme, you will not create any gaps, is also bad. And you have to find the right way in between. Most programs, when you use them, have sort of a default that is done by heuristics, and then people play around with it a little bit. So that's a free parameter.
So to compute this, we will get like a table where the penalties will be written down.
So again, if you run this, if you program that yourself, what you would do is you would look at Wikipedia, ask what is a typical gap penalty, use one once and for all. And for most of the purposes, this will do you fine. And I will get back to more details later on next Tuesday, yes? But sorry, there was another question. I hold up on that. But there was another one here, no? Or did I? He is waiting for longer. Let me.
At the formula, the number of matches is the total number of matches in the whole matrix, or
so this
or just for the
path.
test path?
That's the path. So that's the delta function ij. So here runs the I, here runs the j. So this is essentially the question for one particular superposition of two sequences. You run the I and the j. The question is, do you have a match or not? Delta function, sorry, not sure how familiar you are with the delta function. The delta function is simply, oh, I still have my phone, is simply, this is simply a 0 when it's not the same letter, and it's a 1 if it is the same letter.
No, I get like how do we.
But then one pair.
In this case, it would be 4, the sum, or like 4 and.
No, in this case it would 1, 2, 3, 4, 5, 6, 7. I mean, yes, sorry, yes. The I and j would, yes, the sum would be 4. Correct. Minus the 2 gaps. So minus the penalty, the free parameter times 2. Question over there was?
I was saying that the dot should be above k rather than above k.
Yes, that is correct. So you put the gap here because you essentially put the, yes. Fair enough. Yes. Any other point? But it's the idea there. And we get into the next round where we had this where I posed the question, what is better? So this part here where you have 2 and 2 right versus the one where you have 4 and 7 right. And I said this is not easy to answer. And then we got into scoring matrices. Somebody asked for a Therosaurus. This is not complete, but here are a couple of one. So you can call it scoring matrix. You can call it substitution matrix. You can call it exchange matrix. Log odds matrix. The whole thing in metric, and log odd tables, exchange table, scoring table, yes, all possible variants appear in the literature. And this is one particular one of these, whatever you call it. So here I call it scoring matrix. I try to now use scoring matrix in as many slides as I can. Again, the word doesn't matter, but I try to stick to that word now. And one particular one that is shown here is the Blossom. Forget about the numbers. It's just a name. And in this, the main feature here really is that no longer all the elements on the diagonal, which is in fact an identity match, are equal. Some identities are more important than others, and some non-identities are more important than others, and in some cases the diagonal is smaller than a non-identity match of a biochemically similar amino acid. There are many substitution matrices available, scoring matrices available. Blossom is the most common one, and again there's a series of Blossom matrices, and one is 62. It's compiled by some log odds on some data set of alignments that we know essentially are right. There's an interactive software tool from Francisco Melo that allows you to play around with alignment methods. I liked it when I played around with it. It is, somebody brought up, I'm not entirely sure whether I see that person, somebody brought up a website, Rosalind, that I'd actually never seen before. From essentially, so the group is Pefsner and UCSD. And so somebody in the room here approached me whether I use that. We are not. I asked Lothar to possibly bring that up. I haven't spent much time on it, but from my first looking at it, this is a cool way to look at simple programs that, for instance, tell you how dynamic programming, these alignment methods work. Snippets, you have exercises in there. It's a very interesting website, and maybe in the long run we should use it. I asked Lothar to do it. Maybe when some of you in the room find that this interesting for you, put some pressure on Lothar. Maybe then sort of together you can use it. It looks interesting to me. Okay, let's continue on the pairwise sequencing methods. So the dynamic programming is optimal. but optimal is defined as in I write down the objective function. For instance, the objective function is the delta function that I showed you minus the gap penalty. So that is what I call the objective function, my goal. And given that goal, dynamic programming gives me the optimal solution. That may not be the optimal solution in biology, but there's another problem. There's time problem. The time, again, if I used all possible combinations, compared to that dynamic programming that I call brute force, is immensely fast. But it's still square in the length, right? Now, is that a problem? Do you believe this is a problem? If proteins are maybe 400 residues long, so 400 times 400, is that a big number? And I don't want anybody to give me the answer to what is 400 by 400. I assume you can guess that without machine learning. So is that a number that's preventive? So the algorithm I explained to you, do you believe you can do that for 400 against 400? Who believes we can easily do that on a computer? One. Could we try to have very few people abstaining? Just assume that nobody knows the answer anyway. So what's your gut feeling? So 2 people feel we can do that. How many feel we cannot do that? This is sort of the majority, but the real majority is the abstainers. Hey guys, become active. Move up your hands once in a while, if it is random. No, so 400 against 400 is totally trivial. You can do that on your laptop. You hit return and essentially is there. I do not even know the number, but it's very, very fast. The problem is a different one. And I want to illustrate the problem based on numbers that are sort of realities for us. When I came to Germany, we had in the database about 8 million proteins. So a few years ago, or today, again, I talked about the drop in the data set, but now we're talking about 60 million, actually more than that. Now, in 55 months, this was almost 7-fold increase, okay? This is double every 2 years. This is like computers, right? So for us, in some sense, okay, computers are not really doubling their speed in 18 months anymore, and even 2 years is becoming challenging. The same is true for hard disk space. So 2 years, every 2 years doubling is even in the computer field now a big number, but it's not that big. It's not that scary, right? But now let's look at something else, which is, bless you, is not the number of sequences, but it's the number of comparisons I have to do. And since we have, if we want to do a comparison, what we have to do is run everyone against everyone. So suddenly that's the number we talk about in 29, and that's the number we talk about in 2014. And that's a 20-fold every 2 years. And that really is scary. In the sense, it means that essentially every year now we can do increasingly less to understand or to search for the biodata. So on that scale, and this is, anyway, so on that scale, the number of dynamic programming is way too high. So I can do easily 400 against 400, but I cannot do the same thing many billion times over, or trillion times, 3 trillion times. It is also true that even for the case of the storage that we need in order to keep those alignments, so this is not the same jump because some of the stuff we throw away, but this is still a 5-fold increase every 2 years. And when we came here in 2009, 5 terabyte was a challenge, but you know you could still carry 2 hard disks with 5 terabytes. 300 terabytes is a challenge today. We cannot carry hard disk with 300 terabytes, and you all know that. Just do your backup. I don't know, maybe you have a hard disk that has 4 terabytes. Now you want to back up because it is getting too hot, it's getting too old, something is happening to that hard disk. So you do it backup. This is not the kind of thing you do overnight. And even on the level of machines of LRZ, at some point in between, so we had all our data on some tapes. And at that point, what we tried to do was to, in fact, read off the tapes 50 terabytes. That must have been around about 2011 or something like that. Because we just had a hit on both of our independent file servers. At the same time, they went blank. So we needed to restore some data. How long do you believe it took? And the numbers will not be that different today, to read off tapes 50 terabytes. Do you believe this is doable in a day? No. Do you believe this is doable in a week? So some people begin to be feeling that, some people say no. Do you believe it's doable in a month? So I can tell you, after 3 months we stopped. And we had not even done 1/3 of the data. It is true that our data is stored in a way that is not optimal to be read off tapes. We used tapes because we needed another backup, so we have a lot of small files. And that really was the killer for the tape. But it is something that is really not easy to handle. Okay, the other issue is that we have to choose some parameters. So gap open penalty was a parameter. Which substitution matrix to use was a parameter. But let's turn to a different question. How could we possibly speed up the alignment process? So m by the n, or n square, where n is the length of a protein, I say is too slow. Again, for a single comparison, totally trivial, but on the level of doing that 60 million times over, we cannot do that. So, any idea how we could speed up? Think about fast search algorithms that you know. Think about text searches. How could you speed them up? Yeah?
Building trees?
You could build trees. So this is a true computer scientist. The idea of a cache on a tree, by the way, comes from Munich. I'm not sure whether you know that, from the TUM. But yes, you could build trees and could essentially speed it up by hashing. And that, in fact, is exactly what we do. We have word finds and hashing. One simple way of doing that is shown here. The method is called BLAST, where in the first step, and again, BLAST is not quite doing that. This is slight simplification here. But imagine that in the first step, you search for identical words of length 3. Mat, or whatever, map. I'm blanking because I cannot think about words of 3 because they are so rare in English. No, because my brain is not. Anyway, so here a few of these 3-mer words are found. So this is very easy to map onto a tree, to build my entire data structure into a tree with these 3-mers, and then you can quickly search through that tree to find whether or not in that protein that you're looking at, you find that particular trimer, or that particular K triple, where K is 3. Okay, that part is easy. Now what do you do in the next step? Let's call it, what we do here, we seed the alignment process. So we find identical words and we assume that whenever I find an identical or an important alignment, there must be at least one of these words there. But that's not enough, because so far we replaced the dynamic programming where we have an entire alignment with 2 sequences against little stretches here. And the longer we make the length of the word, the slower it actually gets again. So the speed, the tremendous speed, so 2 is not enough because 2 is too small. Maybe today the database is even too big for 3, but 4, 5. Anyway, so how to extend? Yes, you can, again, you can take a word of 5 or 6, but at some point, when you do this, you will lose, okay, you still have the tree structure, you still gain something, but ultimately, you will lose again.
If you find a match, so you take the ones behind the match.
Okay. So, what you said is, I find a word here, and I'm looking, is there another word match next to it? Is that what you meant?
No, you take what's behind the match, together with the match, and hash it, and
So behind the match is this one here. So the word is E. Oh, my example. Okay, I have to. The example that I show. Here's another mistake. I understand. So again, actually, blast. To my excuse, blast doesn't look for identical words. But maybe for next time I should find a better example. Anyway, so EKV. So EKV and EKA, assume those, for what I said so far, those would both be EKAs. And then what you do, you look at A and V. So you find this one here, and then you go in this direction and look at the next one.
You take a bigger block and hash it.
But taking a bigger block without looking at the smaller block first?
No, you look at the smaller block, because that's where your bigger block starts.
So you begin everywhere to have, essentially have one set of hashes that are small blocks, and one set of hashes that are the continuation of small blocks into bigger blocks. Is that what you suggest?
No, only hashes of bigger blocks, but then
yeah, but that's the problem. So only hashes of bigger blocks, ultimately, well, okay, from 3 to 4 may be better, from 4 to 5 may be better, but at some point it's going to break. It's a break means it's still going to work, but at some point you're going to actually lose because you're beginning to enumerate. So you're beginning to replace the m by or the m by n problem by a problem of m by the 20 by the power of m by the 20 by the m by the n by the 20. So this is a much larger number. So you cannot keep going in making your hashes or your block word size bigger. Okay, let's not debate about 1, 2, 3, 4, 5, 2, 3, 3, 4, 5. But at some point it's going to hit you. And 5, 6, 7, at some point we're getting close to where it actually will hurt you. And we are still, at length of 5, 6, 7, we are still nowhere near typical protein alignments. And when we get near typical protein alignments, which could be 80, then we clearly will be in the hurt category already, so we cannot do that. So how can we begin with these words and make them longer? This time the answer is not through another hashing or through a bigger hash. Any other idea?
Try to start extending from the
How?
Like, I mean, if I have it, how do you have this stored? Like, so you first copy and you have like your hash of 3 or...
Yeah. But again, I believe, just keep going. How would you extend? Don't be too scared about my asking you, but what would be your next step? So extending it. Again, here's the word, and then you have the next one, AV. What do you do? Well, it's clear. If A equals V, you keep going, right? That's the simplest way in which you could do it. You keep going in either direction, and that's sort of illustrated here. You keep going in either direction until somehow you no longer find a match. Or the criterion could be very different. This could be the entire length. So if the average falls below a certain number, or that if the next one is no longer hitting, you stop. There are different ways in which you can play that. Or we could, in fact, try to extend even by dynamic programming, and I'll get back to that in a minute. But whatever that does, the important thing is there will be regions in between where we do not know with this method where we cannot get an answer. Because if the extension ends, so if whatever criterion I used so far says I cannot extend further, and in this one I cannot extend further, and in between there's no word match, then that's it. There is no alignment in between. Okay? Yeah?
I've got a question. How did you choose the 3 words, the 3-letter words in the beginning? Are they chosen random? Or?
No, so essentially what you do is you look at all the triplets that are common between these two sequences. So in the simplest model that I presented to you so far, common means identical words. The. So you simply ask, you simply put a red bar under every the, and you identify the this. Okay, I need to find a better word. The maps. The cogs. Is that clear or I'm not clear about this?
Not really. Because
Yes.
In the upper
Yes, okay. So you say in the sequences that I show they don't.
Yeah.
Yes, I'm sorry, there's an additional problem here. Actually, BLAST does not use identity matches. What BLAST essentially uses is the scoring matrix. It says the scoring matrix that I showed you before, if the value is above some criterion, then I call that a match. And that's why the words that I show here are not identical in letters.
Yeah, no.
But they're okay? I would have to correct that in the next lecture. Again, well, yes and no, because really BLAST does not use identical letters. But I find it, I presented the lie to you because I thought it was easier. Okay, reality is easier. But then it's clear? There was another question? No? Okay, so now we have a new problem. The new problem is that we have to somehow compare, we have short matches, relatively short matches, the way the algorithm proceeds, and we have to compare this, we have to somehow assess what matches are meaningful. Now, meaningful typically is defined as scientifically meaningful. We don't have a feeling for scientifically meaningful really for sequence comparisons. So we have to revert to a much simpler meaningful tool to measure meaningfulness, and that's statistics. So we ask simply, how surprising is a particular match? In order to do that, what we need to do is essentially look at the distribution of the score I get. So I have it begins with a word, it continues for a longer stretch, and I have to ask this particular match, the score of the dynamic program that I had before, subtracting the gaps and all of that. That particular match is here. Although this is actually a bunch of matches here. And the distribution of the random matches in the databases is here. So the degree to which your actual alignment is surprising or is likely to be something that is non-trivial is determined by the difference of the score you observe to the distribution in the background. And it has to do with the height. So the expectation value has to do with the height of the distribution. The height and the spread of the distribution. So that implies that you have the same alignment. So the same, I believe on the previous slides I had some examples. Say you took this pair, this alignment, and you assessed the E-value, the expectation value for that. So essentially how far is the match that you get from summing this value up against using that one against the entire database? How often could you have had such an alignment by chance? Essentially is the question you answer. And now if you're Gaussian, you have the same average, the same sigma, but you have a much, much higher Gaussian, then that means that this hit is more significant. So running the same hit against a larger database will give you an expectation value that is higher. Is that clear for everybody? No? Can I sort of have a more active yes somehow? Is that clear or is it not? Or active no is good too? No, it's not clear. Okay, so what you're trying to measure, so you have a particular alignment in front of you. Let's just talk about a word match. I believe it may be clear with the example that I don't like because I always stumble over it, the. And I ask myself, how frequently do I find the in a dictionary? Or in not a dictionary, in a text, in a newspaper. I look at the New York Times and I ask, how often do I find the word the? And I will very soon realize that finding between two articles many times the does not tell me anything about their similarity. Because the is in every single text, in almost every single sentence. So you have a lot of random probability. So the random probability of finding the is as high as the for the ones that you find. So the red hit that you're looking at would sit completely in the background distribution. So sitting completely in the blue here means that the probability of randomly finding that match is very high. And that's what you want to estimate. The expectation value should estimate how likely is it that this chance that this hit you find, you found because of some important signals like evolution, similarity in structure, similarity in function, or by just random similarities. You have a very huge database. If you look at, we talked about that triplets, typical triplets, so you have 20 amino acids, 20 by 20 by 20 is 8,000. If you run against 60 million sequences, each of these is 400, 500 letters long, then you immediately see that the probability of finding any triplet is very high. It's just because the database is so huge. And that is the point about the height of the curve here. If you look at a database that has 5,000 proteins, then every match, a particular match between the and the, has a higher significance, or in the, has a lower significance if the distribution is lower. So finding the in a sequence, in a database of 5,000 is much more surprising, is much more surprising than finding it in a sequence of 60 million. Because in 60 million you will find it, full stop. The numbers are such. And the difference between these two statements is only the height here. I'm not changing any other parameter. I'm not changing the distribution of this particular T, H, and E in the database. All I'm doing is I just have a larger database, and randomly, if I create my sequences at random, at some point I'm going to create a the. Right?
The question to the slide before this one.
Is it related to this point? I believe this point is still not. I have to try again to explain that point. Can you hold on, or is it related to that point of the e-value?
Somehow, maybe.
That's a tricky one. Is that the one you meant before?
The slide.
This?
Yes, this one. You say the default word size is for seeds is 3,
Mm-hmm.
but in this case, it's you start with 2, right?
No, then there's another problem. No, 1, 2, 3, 1, 2, 3, 1, 2, 3.
Which thing you compared?
No, so my initial hit is here. So this is the so the way the slide builds up is exactly that. So first I look at all the words of 3 that match.
Yeah, but that only matches the first one. The second, the third. And
yes, that is the yes. I'm sorry. Okay, we are back to that point. I'm sorry. No. So yes, I have to do that differently. Essentially, what I'm looking at is this matrix here. When I say match, I don't mean necessarily identical residues. I mean that lookup of these values is above a certain threshold. So I look for words that are not identical, but words that the 3 of them, when I look up in this matrix, have a score that is higher than some threshold. And all of those I call matching words. Is that concept clear? It's very, very important. Okay, but the other part here remains critical. So I have to find a new way of explaining the expectation value. This is a very, very important concept. Not only for sequence alignments, or not only for biology. Again, so you look for, you're asking yourself, you do a text mining, and you wonder whether the word art is a good keyword. Is that something that is descriptive? So the answer to that question, is it descriptive, has to do with how often you find that word. And again, if somebody had come, a non-English speaker had come with a suspicion that this word is a good descriptor, then most of you in the room would immediately shout no, and all of you in the room would immediately shout this is not informative at all. In order to establish whether this is informative, I actually have to look how often do I find that in the type of documents that I have. Now, imagine I gave you 10 newspaper articles, 5 that have to do with something related to paintings, and 5 that don't. And you essentially want to establish on these data, on these articles, you want to establish whether the word art is a good keyword. Essentially, good keywords would be, they would be found in the ones on paintings, but not found on the ones on mathematics or whatever you find, right? And you get a certain hit rate of art in these 5 and in these 5. If I gave you 500 instead, then the hit rate would be very different. Now, assume in 5 you have 100% match, so you only find it in papers about paintings and never in papers about mathematics. Okay, but if in 500, two sets of 500, you have the same result, then you're much more trusting that art is a good word. And essentially you didn't change the word, you didn't change anything about your experiment, just the size of the Gaussian changed. 500 is higher than 5, that's all. And because the height changed and you still observe the same result, that means that your trust in the importance of that result increases. That's the simple fact. And in fact, again, I bet that your intuition is completely aligning with that. I don't believe there's anybody in the room who feels this is shocking or this is surprising. It's trivial. It's just something that you have to rationalize because it's an important feature. So the expectation value, p-value, the probability that some hit arose by chance has to do, importantly, with the height of that. And is essentially a measure for the difference of the score that you observe with respect to the background distribution. And that essentially answers the question, how likely has a particular hit arisen by chance? Okay, now we get into the question of how accurate are alignments, pairwise alignments. So pairwise alignments essentially means you have pairs of sequences that you align. And how can you do annotation transfer? For instance, is it true that similar sequences relate to similar function and similar sequences relate to similar structure? Now, I have this sort of image here of sequence space, so all the space of all the proteins that exist, and that structure space is a little bit smaller because we assume there are more sequences than structures, because we can somehow, we get into that in a minute. So there's a reason for the size. So we typically assume that there are very different sequences that can be closer in structure. And in some sense that is already at the heart why these two circles have different sizes. But we also assume that if two sequences are very similar, they map to something that is recognizably a similar structure. And that is exactly what we talked about last week when the topic was the comparison of 3D shapes. So by the comparison of 3D shapes, we recognize that there's a bunch of those that are essentially similar. And we see that different sequences map to the same. Another important reality is this one here. Most, or many sequences that could exist so that I can create in a computer do in fact not map to any structure. How can that be? Yeah?
Maybe when it's a new structure?
That's it. Okay. So he says, let me redefine, good point. Let me redefine what I'm saying here. So this is really an abstract way of looking at sequence space. I cannot draw it. It's just an image for sequence space. In my image of sequence space, I assume that I know all sequences. And I could draw sequence space. And the same for structure, meaning that it actually, the circle describes all possible structures that I will observe. Maybe not all the ones I know today, but all I can possibly observe if I knew all the structures. So the reason here is not because I add a new structure. So the way I mean this circle really is everything that exists, not everything we know, but everything that exists is actually in there. So this is not because they fold to something that is new. That's not what I mean by that. So how could it be that something doesn't fold to any structure? What does it mean? Yeah? We lose information from going from sequence to structure. Yes, but what I mean here is we have proteins. They are actual beings in some sense, whatever the beginning of life. And so in my image here, a theoretical image, every single one of them is mapped to a structure. Every single protein in a cell has a structure. So how can I still explain that this goes aghast or in the empty? Yes, just 1, 2, yeah?
Sometimes if a protein is denaturated, it cannot hold again.
I get back into that. So the statement here is sometimes proteins are denaturated. Denaturated means they in fact are stretched out. Let's get back into that issue in a moment. There's another point.
Not all sequences lead to a structure.
Not all sequences lead to a structure, which is exactly what the arrows mean, but why?
Maybe that sequence does not exist.
Exactly. So the statement here is essentially, most of the sequences you could create in a computer, you will never observe. They don't exist, and they don't exist because they don't go into the structure space. They don't exist because they don't work. Okay? And actually we believe, it's a little bit complicated to really make that point, but there are reasons to assume that the majority of sequences you can create in a computer will not exist. Let's get back into the denaturated story. So you take a sequence, a protein sequence, you throw it into a solvent, and it adopts a unique three-dimensional structure. That unique three-dimensional structure occupies less volume than the original sequence with which you started. Can you remember the example I gave you, how you have an everyday experience, how you can invert this issue of protein folding? Sequence, solvent, compact, three-dimensional structure. Do you remember the experiment that you can do to invert that? Nobody? That was this class. Okay. No, in this case, so let's begin differently. You all know it. You just have to discover it inside of yourself. And you all, I would challenge, you all have done it. You all have done this experiment. Not in school. I would challenge that many of you have done it over the last half year, over the last month. And there are many in the room who do that every day. Or some in the room who do that every day. Yeah, cooking an egg, exactly. So in the egg, what you have is you have the proteins swim around in a real three-dimensional shape, so condensed. When you cook the egg, you break up the proteins. You actually denaturate them, as this word was. You put them out of their natural structure, their natural habitat, and make it longer. That's why the boiled egg is bigger. That's why it aggregates, and that's why you have a hard-boiled egg. Exactly. But this is in fact denaturation. Now, some proteins in a cell exist in denaturated state due to functional reasons. That is often referred to as protein disorder, and we will talk about that. And sometimes this is just a mistake that comes by you expose the cell to heat or other environmental challenges, and it leads to local denaturation. How do you believe the cell will react to that? So this is not on the level of alignment, but this is on the level of biology. It's an interesting reality. So again, I have in every single cell some proteins that shoot some light on it or some heat, and you will have some proteins that will denaturate. What will the cell do?
I think the cell will explode.
Oh, no. So this is, so one thing about life, it's remarkably surviving. It's remarkably good at surviving. So a little bit of heat, okay, you punch a laser or something, high laser energy, and at some point you will explode it. Yes, but actually, mostly you will explode first the water. But cooking the water, I believe, will be the most of the explosion effect, I suspect. But again, there is an amazing ability of a cell to absorb some heat at least. So what will be the reaction to denaturation? Fusion? Fission of the cell. So you put some heat on some of the cell, and some of the proteins denaturate, and the cell will be so frustrated that it will fuse with other cells, or will break apart. Those are two different ways in which it could be, and that again is an extreme reaction that does happen, yes, but on a lower scale. So I'm not really killing the cell, I'm just giving some heat. So the cell, I don't know, E. coli cells are happy at 37 degrees, I'm going to expose them to 70 degrees or something like that, yeah?
Maybe you can use smaller proteins to deconstruct it.
So maybe is the answer here, it can use other proteins and reconstruct the proteins that it lost. The cell doesn't actually, there are pathways in the cell that would allow something like that. It is true also that you will have a lack of proteins, and sometimes you have a way to sense that and to produce new proteins, but this in fact will be completely anew. So you will just do the same thing that the cell did at its birth, meaning it took the DNA and put it into proteins, and that will just be repeated for those proteins. That will happen. Anything else?
I think a cell has a kind of a trash mechanism, and this will just throw away the...
So here we have one explanation, which is the cell has a trash mechanism. So essentially it has a mechanism for induced kill, or for induced, trash I believe is the better word. But essentially what you have enzymes swim around that eat up proteins that are denaturated and that don't look nice, and you can call it trash. They're really eaten up and digested, put into energy. Yes, something else?
Maybe the heat is like a catalyst for the cell.
Yes, that could be true. Actually, it is true, and there are many cells that actually need the heat for that type of heat for survival. And in fact, there are organisms that need stress to survive. This is also true. That's also a great point. But there's some other mechanism, yeah.
I think that there was something like the cell has a repairing mechanism.
Yes.
I think when it's...
Yes, it does. And they're called heat shock proteins. Just historically, because the first time they were discovered was because people just exposed cells with heat. And essentially you can imagine them as a big basket that is made up of, there are different versions of that, but typically of proteins, there are many proteins in there that each is about 1,000 amino acids long. And typically they have 20 of these. These 20 times 1,000 is a huge thing that forms a basket. And the protein was really sucked into that basket, and then it's repaired inside. How exactly the detail happens inside is still not quite clear, but many things of this inside mechanism of heat shock proteins are clear, and it's remarkable. Because essentially, in fact, this big box rotates to increase the odds, whatever the mechanism, the molecular mechanism is, the odds of, in fact, it's shaking it. Just like a bar mixer mixes a drink, the same thing, the heat shock protein mixes it. It gets a lot of ATP for that and keeps mixing the protein. Out comes a folded protein, and no longer denaturated protein. And in fact, the third mechanism is, and we get back to that, some proteins have to locally be somehow denaturated, not to meet with a trash man, but in fact to meet with regulators. So there could be a signal hidden in the denaturation. Anyway, so we are beyond that point. But many protein sequences that you could write in a computer do not exist at all. That's another reason why this blue one is much larger than the brown one. Now, when we get into the sequence comparison, for pairwise comparison, and what we want to now, our objective B, that we want to compare two sequences and find out whether or not they have a similar structure, from sequence alone. Yes?
But in the previous slide, can you say that the sequence space is constructed by the computer, and the structure space is constructed by analyzing the proteins from the real world?
Oh, that's a good point. I made another mistake in my slides. Point taken. So what she says, and I never stumbled over that, thank you. What she said is that previously when you asked your question, I said this is my assumed entire sequence space and this is my assumed entire structure space. Now I say that actually some of those here are indeed my assumed entire sequence space, but I'm saying they don't exist. Shouldn't I correlate that to a structure space that is also an assumed structure space? Not observation of all the structures that I have, but all the structures that I could have. Point taken. But still, it doesn't end. In my example, then I would make this a little bit bigger, but it still would be smaller than the blue. Sorry? No. But again, so the important thing here is many proteins do not fold to any imaginable structure. So making this larger by sort of, maybe, again, this is a possibility, I do not know. So if you argued there may be structures that we can think about string together in a computer, but we will not observe them. Maybe that's true. We actually don't know that at this point. So there are several people try to do that for particular types of proteins. That they said, well, you know, I look at 4 elements, so 4 helices. And this are my 4 fingers, and I can put them together in different ways. Now just let me sit down with a little bit of bottle, glue and cylinders, and realize all the possible models that I can make, or do it in a computer. And then go into the world of proteins and ask, which one of the ones that I can think about do I find? Efimov was doing that. I remember I was very fascinated reading that 21 years ago when he did that for a particular set of proteins. And I remember that there was a 10 years later paper. So when he did that, for that particular, just a sec, this was called a Greek key architecture. For that particular architecture, he found 32 ways of doing it in principle. And of these 32, he observed 2. 10 years later, half of them had been found. I have not kept up with the story. Maybe the fraction of those that have been found is larger today. Maybe, in fact, all that we can imagine, we will observe. But I believe we're getting too far into yeah?
My question is just if I have this doing it right. You have a protein sequence, and it folds into a structure. And the sequence space is computed or predicted by a computer. Is that right?
So sequence space is a very abstract phenomenon, or abstract image in my head. So the sequence space is for me the space of all the sequences that I have. And let's just forget the computer.
In fact, I don't know the whole space, or do I know everything?
No, because I don't have all the sequences. We currently have 66 million sequences. And what I can tell you, here's another way of looking at it. When I put my hand into the dirt here, and if I would take whatever I find there and sequence it, the vast majority of stuff that I find will not have been seen before. Okay? That same is true for the ocean. That same is true for picking it from the air. People do that in the subway. Barcelona is the last city that has done that, where people just walk through the subway and take wherever they can little bits of material or whatever it is, whatever they find in the subway, and they sequence it and simply ask in the Barcelona subway, how much of the stuff, the living material that I find, has been observed in terms of the 60 million sequences we have. And most of these kind of experiments, so the first one was done in New York on this, and most of what you find you have not seen before. So meaning that the 60 million that we have is by far nowhere near the whole universe.
Okay, but the same applies to the structure space. There are also
correct
structures which are not seen until...
Yes, so now the question is, for the sequence space, we are very confident that the number out there is way, 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 way larger than what we have. For the structure space, how close to saturation are we? And that ultimately depends on how you define similarity in structure. So when you say, okay, this is one object, and it's always the same thing. So these two objects, now you cannot see them anymore, but my story is more consistent. So these two objects, you say they are similar. Essentially they are the same structure. But then you can also say, okay, one is a little bit longer. So it's not really the same. If I call them the same, then can ask, how many new ones did we actually discover over the last 10 years? And there clearly is a sort of point of saturation. It does not currently allow us to estimate how many will we have in 10 years. But over the last 10 years, we have sort of really reached something that looks like a saturation. It may be that, again, tail effect, that the tail is so long that we will have twice, 3, 4, 10 times more than we have today. But today it looks as if saying that it will be half of the whole future may not be that far off. Maybe it's 10. For the sequences, the ratio is way, way, way higher than 10 by the 10. So that is a sort of difference, okay? That image clear so far? Back to that one here. I want to use, so what I'm going to assess now in the following slides is the degree to which I can use pairwise sequence alignments to infer similarities in 3D structures. And it is very clear that there is a region here where this is very simple. So I have a very, very, very similar sequence, 100% say, and they have the same structure, essentially. Again, there's a caveat, there are some issues here, but mostly this is true. So the higher, here what I really measure is sequence identity for simplicity, the higher the identity, the simpler the comparison. I call that daylight zone, because essentially the zone where the comparison is totally easy, in some sense. And then comes the twilight zone, wherever these numbers are. I will get back to where exactly these numbers are, and this is just, I want to illustrate an effect here. So for a high level of sequence identity, I will be able to say something, and then I will sort of lose the signal. Meaning that in that zone here, for some of those, I will from the sequence comparison be able to say, yes, they have a similar structure, but for some I will not. So here the comparison gets more complicated. And then there's a midnight zone in which the overlap between two sequences is random, or on the level of random, and that means I will not at all be able to infer that two proteins are sequenced from the sequence similarity that they are similar in structure. Now when I simply ask about numbers, so what I'm going to do is I'm going to have two data sets, and in these two data sets I'm going to ask what's the probability or the accuracy that if I looked only at hits that are 100% identical, that everything I then find will have a similar structure. Okay, and this is an invented curve here, just to illustrate what I'm going to show you next. So, and the invented curve in this particular case is from beginning here from 35% sequence identity, it goes down to 15%. Again, these numbers are not important. This is just to illustrate an effect here. And the curve is not true either. I just want to illustrate. So, essentially, at a high level of identity, everything you find, every pair that has a high level of sequence identity has similar structure. Again, we have a 3D classification mechanism. By that 3D classification mechanism, we say the structures are similar. And then we go down in sequence identity, and we will find many that do not have the same structure. So this is essentially what I call the twilight zone. At the same time, there's the number of false positives versus true positives. So it's also true, I find the ratio is higher here, so when I stop here I'm making fewer mistakes, but many of those that I want to find are still further down. So going down, yes, I make more mistakes, but going down I also find more things that I want to discover, proteins that have a similar structure, right? So there's a payoff between the red line of true hits, and this again, true hit here means have similar structure, and false hits in red, meaning they have different structure. How could you? But I believe this is the stuff for Tuesday. We are going to continue here on Tuesday.